طلاب طالبات، هذا هو المقطع الثالث من محاضرة الجلايكوجين ميتابوليزم لهذا اليوم. As mentioned in the second part of this lecture, this figure indicates the role of glycogen phosphorylase. This enzyme is the rate limiting enzyme of glycogenolysis that regulate the glycogen degradation or breakdown. The enzyme glycogen phosphorylase affected by glycogen phosphorylase kinase B glycogen phosphorylase kinase A. Glycogen phosphorylase A and glycogen phosphorylase B present as active and inactive respectively and affected by insulin hormone. The other hormone, which are called anti-insulin hormones, including glucagon, epinephrine, required a receptor as indicated in the cell membrane, which stimulate the after receptor binding the hormone stimulate the enzyme adenyl cyclase to produce cyclic AMP. The cyclic AMP is a second messenger, convert protein kinase A, the inactive, into activated form, which then activate the glycogen phosphorylase B إلى glycogen phosphorylase A as indicated in this figure, which is responsible for glycogen degradation or cleavage. هذه مقدمة عن موضوع الجلايكوليسيس glycogenolysis. The glycogenolysis indicate that glycogen degradation consists of 3 steps: the release of glucose 1-phosphate from glucose, the remodeling of glycogen substrate to permit further degradation, and third, the conversion of glucose 1-phosphate into glucose 6-phosphate for further metabolism. This figure indicates the role of glycogenolysis or glycogen phosphorylase. As indicated, we have glycogen alpha-1,4 and alpha-1,6 glycosidic linkage. The glycogen phosphorylase liberate glucose 1-phosphate and then transferase enzyme and glucosidase, which are called debranching enzyme, continue the degradation of glycogen or glycogenolysis. Figure 9, alpha-1,4 glycosidic bond on each branch are cleaved by glycogen phosphorylase, leaving for residue along each branch. The transferase shift a block of 3 glycosyl residue from one side, from one outer branch to the other. In this reaction, the alpha-1,4 glycosidic linkage between the blue and the green residue is broken and a new alpha 1,4 glycosidic bond between blue and yellow residue is formed as shown in this figure. The green residue is then removed by alpha 1,6 glucosidase, leaving a linear chain with all alpha 1,4 glycosidic linkage. الأسفل واضحة هذه suitable for further cleavage by glycogen phosphorylase to liberate glucose 1-phosphate. Glucose 6-phosphate obtained from glucose 1-phosphate has 3 fate. أولا, it is the initial substrate for glycolysis. Okay, هذا هو الرسم وهي التفاصيل. The glucose 6-phosphate derived from glycogenolysis has 3 fate. It is the initial substrate for glycolysis. It can be processed by the pentose phosphate pathway أخذناها بالمحاضرة قبل أسبوع to yield reducing equivalent NADPH with hydrogen and ribose derivatives. ثالثا, it can be removed or converted into free glucose for release into the blood stream. This will occur in the presence of glucose 6-phosphatase enzyme and in liver, and to a lesser extent in the intestines and kidney. The rate limiting enzyme of glycogenolysis is glycogen phosphorylase as indicated. The glucose 1-phosphate formed is converted into glucose 6-phosphate by the same mutase used in glycogen synthesis. Glycogen phosphorylase breaks alpha 1,4 glycosidic linkage as mentioned, releasing glucose 1-phosphate. The figure table 3 indicate comparison between glycogen phosphorylase in liver and muscle. Glycogen phosphorylase in liver activated by glucagon and epinephrine and inhibited by insulin, whereas glycogen phosphorylase in skeletal muscle activated by epinephrine, adenosine monophosphate, calcium الموجود through calmodulin and inhibited by insulin and ATP. Glycogen phosphorylase cannot break alpha 1,6 glycosidic linkage and therefore stops where it near the outermost branch point. The debranching enzyme glucosyl alpha 1,4 transferase and alpha 1,6 glucosidase. هذا الانزيم نسميه debranching enzyme. Remove the glucose from the branched point in the glycogen that have been exposed by glycogen phosphorylase. The two steps process by which this occurs is diagrammed in figure 10. Figure 10 indicates the role of debranching enzyme as indicated. Alpha 1,4 bond nearest the branched point as indicated. Glycogen phosphorylase release glucose 1-phosphate from the periphery of the granule until it encounter the first branched point حتى وصولنا للجلوكوز الأولى. Debranching enzyme hydrolyzed the alpha-1,4 bond nearest the branch point as shown. Then transfer the oligoglucose unit to the end of another chain. Then hydrolysis the alpha-1,6 bond, releasing the single glucose from the former branch. وهذا طبعا مثال رسم توضيحي أيضا الأسفل يكون أوضح بكثير من الأعلى as indicated, non-reducing, and هذا affected by glycogen phosphorylase. حتى النقطة اللي هي alpha-1,6 يحتاج إلى debranching enzyme as indicated. تلاحظون glucose تخرج as glucose 1 phosphate، وعندما تبقى 4 مجاميع للجلوكوز 3 مجاميع تنتقل، ثم المجموعة الأخرى تحتاج إلى glucosidase activity. أوكي، وأخيرا unbranched alpha-1,4 polymer substrate for further phosphorylation action. Glycogen phosphorylase exists in 2 different forms as mentioned A and B, figure 11. Regulation as mentioned previously في الفيجر المتعلق بالهرمونال effect. هذا steps of glycogenolysis أخير يوضح لك كيفية حدوث الكليفج by alpha-1,4 glycogen phosphorylase، ثم انتقال 3 مجاميع بالوسط يتكون مجموعة طويلة straight chain وبها only 1 glucose مرتبطة بالألفا-1,6 تحتاج إلى debranching enzyme، وبالتالي يتكون straight chain of glucose polymer. إلى اليمين هذا affected by glycogen phosphorylase ينتهي بتكوين glucose 1 phosphate. The defect in glycogen, enzymatic defect in glycogen metabolism lead to various accumulations of glycogen intermediate in one or more tissues، وهذا lead to ما يسمى بالglycogen storage diseases. الفيجر هذا الأخير يوضح لك، فيجر 12 يوضح لك كيفية glycogenolysis. glycogen storage diseases يوضح لك أنواع مختلفة من الأمراض المتعلقة بالإنزيماتيك defect which lead to various ما يسمى glycogen storage diseases. Type of glycogen storage diseases: von Gerk disease. ما هو الديفكتيف انزيم؟ هو جلوكوز مثلا 6 phosphatase or transport system. الأورجين affected glycogen in affected organ increased amount normal structure إلى آخره. و clinical features: massive enlargement of the liver, failure to thrive, severe hypoglycemia, ketosis, hyperuricemia وhyperlipemia إلى آخره. pump disease والإنزيم defective والأورجين affected وإلى آخره. هذه بعض ما يسمى بالglycogen storage diseases with various details. شكرا جزيلا، وهذه آخر محاضرة، آخر مقطع لمحاضرة الجلايكوجين ميتابوليزم، والسلام عليكم.